Good morning, everyone. Good morning, guys. Good morning, everyone. Good morning, people. Good morning. Am I audible and visible to you all? It's live. Good morning, people. Hello, everyone. I hope I'm audible and visible to you all. So today we will continue with the topic blood. Yesterday we started with the blood topic. We completed the plasma part. Today we are going to talk about the formed elements. Okay. So yesterday when we started with blood, we discussed that blood has two components, blood plasma and formed elements. We discussed the blood plasma. Blood plasma makes up the 55% of your blood and 45% is made by your formed elements. In blood plasma, we discussed 90 to 92% is water, 6 to 8% are protein, and remaining all nutrients, waste, hormones, vitamins, they are present in the blood plasma and along with the minerals. And then we have the formed elements, right? And after that, we also discussed in plasma 6 to 8% plasma proteins. The important plasma proteins such as albumin, fibrinogen, and after that we discussed the globulin as well and prothrombin. So this is what we discussed in the last class. Today we are going to talk about what? Today we are going to talk about formed elements. Okay. So I hope yesterday's class was clear. Now we will go ahead with the formed elements. So just confirm in the chat box, guys, I'm audible and visible to you all. In the chat box, guys, quickly, students, confirm I am audible and visible to you all. Okay, I am audible and visible. Very good. Superb. So let's go ahead. So today's topic is formed elements. Today's topic is formed elements. Okay. Formed elements. So formed elements are basically the cellular part of the blood. Okay. Which are the different cells or part of the cells which are present in the blood. So basically there are three types of formed elements. Formed elements can be also called as blood corpuscles. They can be also called as blood corpuscles. Formed elements can be also called as blood corpuscles. There are total three types. First is the erythrocyte, that is your red blood cell. Then we have the leukocyte, the WBCs, and then we have the thrombocytes, which are the platelets. So which are the three, guys? RBCs, erythrocytes. Then we have the WBC. WBCs are known as leukocytes. Okay. Leukocytes. And then we have the platelets, which are called as the thrombocytes. One by one, we will see. And what is the importance of all these three type of cells and what function they are doing, we have to cover it. Okay. So let us go ahead and first talk about RBC. Let us first talk about what, guys? RBC. RBC is also known as red blood cell. RBC is also known as your red blood cell, or the another name is erythrocyte. Erythro basically means red color and cyte meaning your cell. Now, they are the most abundant of all cells in your blood. They are the most abundant. So out of the three, RBC, WBC and platelets, the most abundant number, the highest number will be of RBC. So how much is the number? Number is very important. Okay, so if you are doing a blood test, in blood test, you should know what is the normal range of RBC, WBC and platelets. Because this blood test, blood report can tell you a lot about the patient status. Okay, so what is the normal number of RBC? A healthy adult man has roughly 5 million to 5.5 million per millimeter cube of blood. Per millimeter cube of blood. So 5 to 5.5 million is necessary, absolutely necessary in your blood. If the number goes down, If the number goes down, if the RBC number goes down, but obvious, the hemoglobin will be coming down and the oxygen carrying capacity will be decreased. Your blood will be not able to carry a lot of oxygen. Okay? And if oxygen is not enough in our body, automatically you will not be able to do the breakdown of the glucose, because glucose breakdown requires oxygen, and that will give you ATP for your many metabolic processes. Okay, so the person will get tired, weakness, fatigue. Okay, all that symptoms will start coming up. So normal number is 5 to 5.5 million. So in your home also, if some person has done blood test, you can see the RBC and the normal range they have given. Different diagnostic test centers have different range, but as per NCRT for your exam, you need to remember in a millimeter cube. In a millimeter cube, there is 5 to 5.5 million RBC. Now, how much is a millimeter cube? How much is 1 millimeter cube? How much is 1 millimeter cube? How much is 1 millimeter cube? Just one tiny drop of blood. One small tiny drop of blood, which roughly occupies the volume of 1 millimeter cube, will contain 5 to 5.5 millions of RBC. Okay, one tiny drop of blood is 1 millimeter cube. In that we are having 5 to 5.5 million. Now you can understand, we have 5 liters of blood. You can convert the liter into millimeter cube. So you can get the total exact number that how much RBC do we have. And very interestingly, when you go to medical college, you will have a small practical as well. Okay, where you will doing the practical on yourself. You will be taking a small, needle-like object, piercing your finger, and then putting that blood on the special slide which is available to you. And in the microscope, you are going to count the number of your RBC. So there is a special formula. You don't have to count the whole 5 to 5.5 million RBC. Okay, there is a special formula for it. You have to just count few of the RBCs and then you have to apply it, and then you can get the number if your RBC number is normal or not. Same you have to do for WBC as well. Okay, so that is the number. Okay, now where is the RBC formed? Where is the RBC formed? RBC is formed in the red bone marrow. Red bone marrow in the adults, which are having the stem cells to produce the RBC. So very important point, RBCs are formed in the red bone marrow. Now, this is for adults. This is for adults. So especially your long bones, in the long bones, you have the red bone. Now this is for adults. For adults, you will mark red bone marrow. But if I ask you for a fetus, unborn baby, where you will say the RBC is formed for a fetus? Because in fetus, a baby who is not born yet, they are not having that bone development. They don't have the red bone marrow yet developed, where they will be formed. No, Kinvesh, that is not the answer. Fetus, where will the RBC will be formed? Fetus, very good, Neha Gandhi. Spleen and liver. Their answer is liver and spleen. Okay, so just make a small note of this. In fetus, because see, in child, there is no yet development of the bone marrow. In fetus, there is no development of the bone marrow yet. So there should be another place where the RBC is formed, that is the liver and spleen. But as the child is born, as the child is growing slowly, the bone marrow develops, and then the bone RBC synthesis shifts. It shifts from the liver and spleen into the bone marrow. Okay? Are we clear, guys? ठीक है? Okay? That's your answer. So for adults, you will mark red bone marrow. For fetus, you will mark liver and spleen. Are we clear, guys? Are we clear? Is this slide clear, guys? Just give a thumbs up, guys. Is this slide clear to you all? Yeah. Very good. We shall move ahead. Now we will talk about the RBC structure. Sir, in all adults, do spleen and liver lose the capacity? Yes, beta. Spleen and liver do not produce RBC in adults. That is just the bone marrow, red bone marrow. Moving ahead. Now we will talk about the RBC structure. Look at the RBC structure here. Okay. So red blood corpuscles of all adult mammals are enucleated. So many of you might knowing that our RBC doesn't have a nucleus. Our RBC of adult mammals are enucleated, means no nucleus. Nucleus is absent from RBC. So during the formation stage, in the bone marrow, during the formation stage, nucleus is there. The RBC, when it is developing, the nucleus is there. But when it is getting matured in the bone marrow, the nucleus is removed. Now tell me why the nucleus is removed. Why our RBC is lacking nucleus? Why it doesn't have the nucleus? Come on. Why it doesn't have the nucleus? Yes, very good, Ajay. No nucleus, no mitochondria, no Golgi apparatus, no endoplasmic reticulum. Not only nucleus is absent, no Golgi, no mitochondria, no endoplasmic reticulum. All these organelles are missing. Why? Why we have removed the organelles? Why we have removed the nucleus? Yeah, to increase the space inside the RBC cell, because we need to carry oxygen, and the protein which carries oxygen in the RBC is hemoglobin. So for making more space to hold hemoglobin, we have removed the nucleus, we have removed the mitochondria, we have removed the Golgi, we have removed the endoplasmic reticulum. Okay, so lot of space will be there for, what, guys? Will be there for hemoglobin. Okay. Next, further, there's biconcave. If you see the top view, if you see from the top view, it will appear circular. If you see the RBC from the top view, it will be circular. But when you take a cross section, the side view, if you notice the side view, it will show biconcavity. What it will show? Biconcavity on both the side. Okay, so it is biconcave in the side view and circular in the top view. Biconcave in the side view in a cross section, and in the top view it will be circular. Circular, understood, that is a cell, so it will be circular. Why it is biconcave? Biconcave surface helps in increase in surface area. Increase in surface area for gas exchange. So basically your RBC is carrying hemoglobin inside. Hemoglobin are transporting oxygen. So first of all, you remove the organelles to keep more hemoglobin. Hemoglobin are going to carry the gases, oxygen and CO2 both. Okay, hemoglobin can carry both CO2 and oxygen, and these gases need to be exchanged with the tissues and the alveoli at the lungs. At the lungs, there will be exchange of O2 and CO2, and the same gases exchange will occur at tissues, vice versa. CO2 from tissues to RBC, and from RBC, O2 will go to the tissues. So for gaseous exchange, you require a big surface. So quick exchange can occur. So biconcave provides a larger surface area for exchange of gases, and also this biconcave structure helps to pass through narrow capillaries. Through narrow capillaries. So some of the capillaries, you might be knowing that capillaries, blood capillaries are very thin. Blood capillaries are very thin. So to pass them, the biconcave surface helps. So there are two functions of biconcave: to increase surface area for gas exchange, O2, CO2, very fast. And then this biconcave structure can easily squeeze, easily, flexibility is increased of the RBC membrane, and then it can easily pass through the blood capillaries. Okay? Are we clear? Are we clear, guys? Sir, if there is reformation in the RBC, will it lead to mental problems? It will not lead to mental problems, but other problems may occur, like in sickle cell anemia, thalassemia, the structure is damaged. So, sir, what happens if hemoglobin is absent? Hemoglobin is absent and the person will die, na? Because there is nothing to carry the oxygen, right? Sir, then how do they function without the cell organelles? Good question, Swarup Reddy. You might be knowing there is a metabolic process called as glycolysis. In glycolysis, there is no need of oxygen. Without oxygen, the glucose is broken down in the cytoplasm, and roughly 2 ATPs, net 2 ATP is released, and that 2 ATP can be utilized for the whatever metabolic process, that is the gaseous exchange happening in the RBC. So aerobic respiration is not happening in RBC. Okay, so very important point I'm telling you. RBC, since doesn't have the mitochondria, it carries the oxygen, but it cannot use the oxygen because there is no mitochondria. So it will be doing glycolysis. Okay, and then with the glycolysis, they are getting the energy. Anaerobic respiration they are doing and gaining the energy. Right? Are we clear, guys, with this? Next. So biconcave shape has what? Advantage in gas exchange, as it has more surface area than a sphere. Next, one more important point to note. Camel. Camel is a mammal in which you can see some of the RBC may show nucleated RBCs. Camel and llama. Camel and llama are the two animals in which you may rarely observe nucleated or oval RBCs. Very rarely. Very rarely we can see in camel or llama some nucleated RBC present. Mostly their RBCs are also enucleated majorly, but few RBC may show nucleated presence. Okay, right? Moving ahead. Up now, these were the points of RBC. Now, what is RBC having? Come on, very important. All that space. Why we made all that space? RBC can only pass through capillary. No, beta. RBC passes through all blood vessels except the lymphatic vessels. RBC passes through everything. Artery, vein and everything. Yeah, but capillary is very narrow in diameter. So there it requires the biconcave structure to pass. Okay. Now all that space we created in the RBC was to carry hemoglobin. Right? You removed all the organelles from the RBC, you created a lot of space to carry hemoglobin, because hemoglobin is the protein, is the respiratory pigment in the RBC. It is a pigment because hemoglobin gives color also, the red color. So it is pigment also. Plus it is responsible for carrying the gas. So it is a respiratory pigment. How much hemoglobin we should have? What is the normal number of hemoglobin? Okay. So as per the NCERT data, different books may have different data, different reference ranges, but we have to follow NCERT data. So what is the data given? 100 ml of blood, 100 ml of blood in a normal healthy individual should contain 12 to 16 grams of hemoglobin. Okay, so this is also a very important blood test. So if you have some blood test report at your home, you can just have a quick look. You can look at the number of RBC, and then you can also look at the number of hemoglobin. Okay. So hemoglobin number is how much? 12 to 16 gram of hemoglobin. Normally in females, it is little lesser. It is found usually female are suffering from less hemoglobin presence in their RBC. That is due to menstruation. Okay, in females, mostly they go to menstruation or the menstrual cycle in which there is a loss of blood every month. Okay, even though it is a little bit loss, but still lot of hemoglobin is lost. So you may find females mostly suffering from anemia. Okay, anemia is a condition where the hemoglobin or the RBC count is very low. Now, anemia is a very tricky disease. A lot of Indian females and nowadays males also are suffering from anemia. A lot of population is anemic because lack of vitamin B12 in the diet, lack of iron deficiency, anemia is there. So, a lot of factors are there. Most of the factor is the iron which is lacking in our diet. So, iron deficiency, anemia may occur, or vitamin B12 that is also required for the maturation of RBC. If that is not there, then again a different kind of anemia, megaloblastic anemia may occur. So anemia is a condition where the hemoglobin number is very low, and most of the people are Indian suffering, especially females are more suffering as they are losing bit of blood in the menstrual cycle. So we need to take care that we should have enough vitamin B12 and iron in our diet. Are we clear, guys? Now talking about the lifespan of RBC. What is the lifespan of RBC, guys? RBC can only survive up to 120 days. Okay, after 120 days, the RBC gets tired. After 120 days, the RBC gets tired. Okay, after 120 days, the RBC will get tired. So when RBC gets tired, we have to break it down. So lifespan is 120 days, and after 120 days, the RBC will be sent to the graveyard. RBC's graveyard is spleen. What is the graveyard of RBC, guys? Spleen. So in the spleen, on the left hand side, roughly here, in the little upper abdominal segment on the lateral side, we can say the spleen is present. Okay, and spleen will destroy the RBCs which have completed 120 days. The macrophages are there, the WBC macrophages are there. They can recognize the old worn out RBCs. So what happens due to so much of exchange of gases, the RBC surface, the membrane gets damaged, and then it is not that useful in gaseous exchange. So what we do, we destroy the RBC. Okay, so after 120 days it is destroyed. Slowly, kuncham, little bit of RBC is destroyed. Okay, sir, what role does hemoglobin play in oxygen support? In oxygen transport, hemoglobin only carries the oxygen, na Mohammed? Single molecule of hemoglobin can carry roughly 4 molecules of oxygen. So one hemoglobin structure can bind to 4 molecules of oxygen as well as CO2 also. So many people just know that hemoglobin can carry or transport only oxygen, but it can also transport O2 as well. Okay, come on, moving ahead. So hemoglobin, see, read, they play a significant role in transport of respiratory gases. Old and damaged RBC are destroyed in the spleen, and spleen is known as RBC's graveyard. Very, very important function. Okay, very, very important function. So are we clear with this, guys? Are these points clear? Is this point clear? Sir, what should we eat to obtain vitamin B12? Eggs and milk. Eggs and milk are the very good source of vitamin B12. Okay. Eggs and milk, they are very good source of vitamin B12. We can have that. Moving ahead, guys. Okay. Now we are done with RBC. Come on. Little bit of quiz, little bit of questions. Okay. So yesterday we discussed plasma protein. Come on, let me see how many people are alert here. Okay. The plasma protein which is responsible for blood coagulation. Globulin, fibrinogen, albumin, lymphocytes. Which is the plasma protein responsible for blood coagulation? Come on, quickly. Which is the option? A, B, C, D. You can also consider it as 1, 2, 3, 4. Quickly, guys. Hardly 5 seconds are required for this question. So, globulin is also a plasma protein, but that is required for antibodies, right? The immunoglobulin, so that is not our answer. Albumin is required for osmotic balance, right? So that is also not responsible for blood coagulation. And lymphocyte is a kind of a WBC. It is not also a plasma protein. So only one option is left, that is option number 2, that is option B, fibrinogen. Fibrinogen is the plasma protein required for blood coagulation. You tell me, fibrinogen is in active state or inactive state in blood plasma? Is it active or inactive in blood plasma? In which state it is? When blood coagulation is not required, it is in which state, guys? Very good. It is in inactive state. Superb. Very good. Very good, guys. Superb. Next question. Shall we take one more question or shall we go ahead? You need one more question? Shall we discuss one more? Let's discuss one more question. Come on. In males, today's topic question, in males, the number of RBC, that is red blood cell, in 1 cubic millimeter of blood is how much? In 1 cubic millimeter is how much? In males, the number of RBC is how much? Very good. So clearly you are marking answer A. Very easy question. 5 to 5.5 million RBC. 5 to 5 million RBC. Let's quickly do one more question. Let's quickly do one more question. Come on. Match the column. Very important. Match the column question. Match the column question, guys. Match the items column 1 with column 2. Very important question. Fibrinogen, globulin and albumin, you have to match. These are the plasma proteins and these are their functions. You have to match with the functions. Come on. So what will fibrinogen will do? Fibrinogen, just now we studied. Fibrinogen is a blood clotting factor. So fibrinogen will be blood clotting factor. A will be 2. Globulin will be for defense mechanism. B will be 3. And albumin will be used for osmotic balance. Very good. Super. So what is the answer now here? A is 3. And A is 3 only, your albumin C is 1. C is 1, okay, C is 1 here also, here also. Now literally see A. What is A answer? A is 2. And A is 2 only in B option. A is 2 only in one option. So such questions are expected in NEET exam. In fact, it might be a PYQ also, okay. Probably this plasma protein functions are asked in the previous NEET exam as well. Okay. So this is our answer. Okay. Now quickly. Okay. So I can see many of you are awake. Now we will go with WBC. Let us talk about WBC. Come on, guys. Shall we go ahead? WBC. Come on. So WBC, they are also known as white blood cells. WBC, they are also known as white blood cells or leukocytes, because leuco means white. Leuco means white. Okay. Like in your RBC, it was erythro. Erythro means red color. Here, leuco means white color. White blood cells. So, because they are colorless. They don't have any respiratory pigment. They don't have any respiratory pigment. Lack of hemoglobin. So, when you do RBC counting, like when you go in the MBBS first year in medical college, in the physiology, I think subject, you will do the practicals. And there, when you do the RBC counting, during RBC counting, you don't need to put any stain. You don't need any stain because RBC itself is colored. That will be easily visible. But if you are doing the counting of WBC, you have to put some stain. A stain is available. That stain you need to put on the slide. That stain will stain the granules and the nucleus present in the WBC, and then you can visualize which are the different WBCs, and you can do the counting so that you can understand the number is normal or not. Okay? So they are colorless, but they are nucleated. So a lot of difference with respect to RBC. RBC was colored. Here it is colorless. RBC was enucleated. Here WBC is nucleated. But the number is relatively lesser. Number is relatively lesser. Number is relatively lesser. RBC was 1 millimeter cube. RBC was 5 to 5.5 million. Here 1 millimeter cube will have how much WBC? 1 millimeter cube will have 6,000 to 8,000 millimeter cube of WBC. In 1 millimeter, so in one tiny drop of blood, there was 5 to 5.5 million RBC. In same tiny drop of blood, how many WBCs are there? 6,000 to 8,000. Okay? 6,000 to 8,000. Now, if someone has infection, if someone's body is having any infection, okay? So what do you think? WBC number should normally increase or decrease? Come on, tell me. Come on, guys. If someone is having infection, then RBC number kya hona chahiye? What should the RBC number happen? The RBC bolo, WBC number. If someone has an infection, then WBC number should increase. Yes, absolutely. So you may see 10,000, 11,000, 12,000. The WBC number will increase. That condition is called as leukocytosis. That condition is called as leukocytosis. So just by looking at the blood report, okay, the blood test is called as CBC, C.B.C, CBC, complete blood cell count. In that you will get the RBC number, in that you will get the hemoglobin number, in that you will get different WBC number and platelet number. So overall the count of WBC in a normal person should be 6,000 to 8,000 per millimeter cube. This range may change. This range may change. This range may change. If the person is having infection, the WBC number may increase, right? And if it is a blood cancer patient, you might be knowing in blood cancer, leukemia. Leukemia is a type of blood cancer where the number may go to 1 lakh per millimeter cube. How much is the normal number? 6,000 to 8,000 per millimeter cube. In leukemia, it's a blood cancer where you can see the WBC is going 1 lakh per millimeter cube. So don't think that the person will be having high immunity. Sir, 1 lakh WBC per millimeter cube, the immunity is increasing. No, that WBCs are useless. Immature WBCs. Okay? Immature WBCs. So that all part we are going to study when you come offline. So a lot of interesting things are there. Okay? Then RBC's shelf life was how much? RBC's shelf life was 120 days. But leukocytes are generally short-lived. Leukocytes are generally short-lived. 10 to 15 days. Generally short-lived. They have a very short life. 10 to 15 days. Except memory T and B cells. Memory T and B cells. So if you remember in the acquired immunity topic, I told you that once we interact with a pathogen, foreign body, we generate a memory. So second time when the same foreign body attacks, using the help of memory from where? Memory from T lymphocytes and B lymphocytes. Using that memory cell, we quickly destroy it, recognize and destroy it. So memory cell have a lifespan of about 15 to 20 years. So memory cell will have 15 to 20 years of lifespan, but the normal leukocytes roughly have 10 to 15 days. In fact, some WBCs are there, they just have a lifespan or 1 or 2 days. Okay, so in general 10 to 15 days, but memory cells will have more, right? And now my question, where is WBC formed? What happens when WBC count decreases? Yes, Shinchan, you are correct. In the case of bone marrow suppression, okay, there is a disease where bone marrow suppression may occur, or there are some infections where the WBC counts drop downs. Okay, there are certain infections where WBC may drop down. Okay, that is leukopenia condition. So it may happen as well. My question was, where are the WBC formed? Same answer, red bone marrow. RBCs are also formed in the red bone marrow. WBCs are also formed in the red bone marrow. Okay. Sir, when does the offline class start? I think it will start in the first week of June, beta. How can person improve his white blood cell count? Just have good diet, beta. Have good diet, that's it. Good diet means proper, enough protein should be there in the diet. Carbs less, protein more, automatically. Sir, what are the disadvantage of bone marrow suppression? Bone marrow suppression is a very dangerous thing, especially when a cancer patient goes under chemotherapy or radiotherapy. Especially in the chemotherapy and radiotherapy, when the body is exposed to very strong chemicals or the radiation, the bone marrow may get suppressed, and in that case, the WBC counts may go down. So at that stage, you can see the number has went down. But after the chemotherapy or radiotherapy is done, again it bounces back. Again the number will come back to normal. Chalo guys, I will go ahead. Okay, we have a class to take. So next, types of leukocytes. Come on guys, quickly. Very short time is left. Lot of things to say. Types of leukocyte. There are two types of leukocytes. Granulocytes, agranulocytes. Now what is the meaning of granulocyte and what is the meaning of agranulocytes? So granulocyte, two types of leukocytes are there guys. Granulocytes and agranulocytes. Granulocytes, agranulocytes. Now in granulocytes, granules are present in cytoplasm. What are present? Granules are present in cytoplasm of the WBC. Okay? Whereas in agranulocytes, granules are absent. Granules are absent in cytoplasm. Okay? Now what are these granules? These granules are basically different chemicals which help us to destroy the pathogens. Different chemicals are there in these granules. Okay, different enzymes, very strong enzymes which can kill. Some enzymes are there, some proteins are there which can kill and digest the foreign bodies. So some WBC have this. They are called as granulocytes. As you can see, neutrophil, eosinophil, basophils, they are granulocyte because they have granules in them. Agranulocytes like monocytes and lymphocytes. Monocyte and lymphocytes are agranulocyte. Now why these are called as neutrophil, eosinophil, basophil? So let's see one by one. Like if you see neutrophil, phil means to like. Phil means to like here. What is the meaning of phil? Phil means to like. Neutrophils are having neutral staining granules. What they have? Neutral staining granules. Eosinophils have acidic staining granules or eosin staining. Acidic staining or eosin staining. So eosinophils can be also called as acidophils. Basophils are having basic staining granules. Look here guys. Okay, I will take the use of a board. That will be much better. Or else you will get confused. Granulocytes. Granulocyte is a type of a WBC. Which one is the first one? First one is the neutrophil. Neutrophil. So here is the WBC. Okay. Now they have what staining granules, guys? They have neutral staining granules. So in the cytoplasm, they will have these granules, and these granules stain with a neutral stain. So there are three types of stain: neutral stain, acidic stain, basic stain. So here the granules are getting stained by neutral stain. Okay. And next, talking about the nucleus. The nucleus is multi-lobed nucleus. What is the nucleus? Multi-lobed nucleus. Sometimes three-lobed. So this is the nucleus, guys. What is this? This is the nucleus. Here nucleus is multi-lobed. Multi-lobed. From 3 to 5 lobes. How many lobes? 3 to 5. Right now you are seeing 3 lobes. Sometimes you may observe 4 lobes, or sometimes we may also observe 5 lobes. How many lobes? 5 lobes. So this is your neutrophil. Maximum type of the nucleus is lobulated beta. Nucleus, normally when we draw a cell, a normal cell is like this. Normal cell is like this. Here you have the cell, here you have the nucleus, right? Here is the nucleus. But here the nucleus is multi-lobe. The nucleus is divided into lobes. Okay? So 3 to 5 lobes are present in neutrophil. Quickly we will see next cell. Next cell is eosinophil. Next cell is which one? Eosinophil. Okay? Now notice eosinophil. Eosinophil in the cytoplasm, they will be having acidic staining granules. Which granules? Acidic staining granules. So you can see the color is little reddish in color. So these granules are here acidic staining granules. Which granules? Acidic staining granules. And nucleus will be bilobed. Nucleus will be what, guys? Nucleus will be bilobed. There are two nucleus. Two-lobed nucleus or bilobed nucleus. Two-lobed nucleus or bilobed nucleus. Okay? So eosinophil will be bilobed. And granules will be acidic staining. So very interestingly, so when you are observing in the microscope, if you observe multi-lobe nucleus, you will count it as a neutrophil. If you observe bilobe nucleus, you will count as a eosinophil. And then the last type will be basophil. The last type in the granulocytes will be the basophil. Baso means basic stain loving. Okay, so here is the basophil. Basophil love basic stain. Which stain? Basic stain. And basic stain color is blue. Basic stain color is blue. Okay. So in the cytoplasm there is basic staining granule. Okay. Basic staining granules, and the nucleus will be twisted. The nucleus will be what, guys? The nucleus will be S-shaped or a twisted nucleus. Or I will say what nucleus? The nucleus will be twisted. क्या कहेंगे? Twisted nucleus. So in basophil there is twisted nucleus. So if you notice, in all the cells granules are present. So granulocyte is a type of WBC which show presence of granules in the cells, and these granules are different chemical proteins having enzymes responsible for a particular function. And based on the color of the granule, based on the shape of the nucleus, we can identify 3 types of granulocytes: neutrophil, eosinophils, and basophils. In neutrophil, multi-lobe nucleus, sometime 3, sometime 4, sometime 5 lobe nucleus. The granules are staining with neutral stain. Eosinophil will have bilobe nucleus, acidic staining granules. Then we have the basophil. Basophil will have twisted nucleus and basic staining granules. These are your what? Granulocytes. Okay. Now, which are the next group? Which is the next group, guys? After granulocytes comes what? After granulocyte, the another group of WBC is agranulocyte. Which side? Agranulocytes. Achha, going here. Another name of eosinophil. Eosinophil has acidic staining granules. So eosinophils can be also called as acidophils. Can be also called as acidophils. Are we clear? So what is the use of nucleus, beta? That nucleus helps to synthesize the proteins. Nucleus contains the gene. It helps to synthesize certain proteins which are constantly required by the WBC. That's why the nucleus is present. Okay. Next, agranulocyte. Now agranulocytes are of two types. What are the two types? The first one is the monocyte, largest of all the size of WBC. Monocyte is the largest of all the WBCs in size. And then we have the lymphocytes. Lymphocytes, which are mostly present in the lymph. So we call them as lymphocytes. Now monocyte, as I told you, they are the largest of the WBC. Their size is largest and their nucleus is kidney-shaped nucleus. What shape nucleus? Kidney-shaped nucleus. Kidney shape, or I can say horseshoe shape. Kidney-shaped / horseshoe-shaped nucleus. Horseshoe-shaped nucleus. And lymphocytes are the smallest in size. Smallest size. Okay. In lymphocytes, little smaller size. And nucleus will occupy the majority of the portion of cytoplasm. Nucleus, if you see, the nucleus occupies a very large portion. The nucleus occupies a very large portion. This is the nucleus. So nucleus is very large, almost occupying the whole space of the cell. And lymphocytes are of two types, T lymphocytes and B lymphocytes. T lymphocytes and B lymphocytes. So are we clear with the types, guys? If we are clear with the types, we will quickly talk about the functions. We will quickly talk about the functions. So let us see one by one the functions now. Okay? Come on. So first one is neutrophils. Come on, guys. First one is neutrophils. Pay attention. Neutrophils are the most abundant of all the WBCs. So if you see, the most highest number of WBC is neutrophil. 60 to 65% of total WBC is neutrophils. So wherever any infection, any injury happens, the first one to report at that site, which will be that WBC will be neutrophils. Like if some problem happens in our community, some crime happens, so we call the police and quickly some police comes with the van. So similarly, which police will come first? Any injury or infection in your body, any injury or infection, the most common WBC to report at that site will be neutrophils, because neutrophils are the most abundant cells. Okay, the nucleus is multi-lobed. The nucleus is multi-lobed. They are phagocytic cells. Phagocytic cells means they will come, they will eat, they will engulf and eat and digest the microbes. They will eat the foreign organisms because they are phagocytic cell. So any infection in your body, any foreign body comes, neutrophil will come and try to digest it. Okay, you might have seen pus formation, guys. Sometimes you may have got some pimple or some boils on your skin. You might have seen some white pus coming out from there, and that white pus is nothing but what? Nothing but neutrophils. Nothing but what? Dead neutrophils. Yeah, I know, guys, the time is up, but we started little late, so I may extend 5 minutes, right? So as you can see, you're wasting time only, majority of you on the online class. So I can extend a little bit, 5 minutes, okay? So please bear with me. Next, basophils. Which is the next cell, guys? Basophil. Basophil are how much percent? Basophil are how much percent? Basophil is the least. I hope you are noting down as well. Neutrophils are 60 to 65%. The least number will be of basophil. Basophil is only 0.5 to 1%. Basophil requirement is very less in our WBCs. What do basophils secrete? Very, very important. Basophils secrete histamine, serotonin, heparin. Basophil secrete histamine, serotonin, heparin, which are involved in inflammatory reactions. Which reactions? Inflammatory reactions. So if you notice, if you get any injury, if you get any cut, you can see redness, swelling, pain in that area. The redness, swelling and pain in a particular injured area or infected area is called as inflammation. What it is called as? Inflammation. And which cell is responsible for inflammation of the WBC? Basophils. Which cells? Basophil will be responsible for inflammation. So anywhere in your body inflammatory reaction is happening, either inside or outside the body, basophil will be responsible. Moving ahead, next cell, eosinophil. Which is the next cell? Eosinophil. You can see the granules are red in color, acidic staining granules. The nucleus is bilobed. The nucleus is bilobed. How much is the percent? Of total WBC, how much eosinophils are there? Out of total WBCs, eosinophil are 2 to 3%. How much percent? 2 to 3%. And they also resist infection. You can note down, they resist mostly parasitic infection. They resist which infection, guys? They mostly resist parasitic infections. Parasitic infections. Parasitic infections they will resist, and they are also associated with allergic reactions. They are also associated with allergic reactions. Okay? Resist towards parasite. Parasite means like if you get some worm infection, Escherichiasis, filariasis. When a person gets worm infection, in most cases, eosinophil number increases. Eosinophil number increases, okay? And it also causes the allergic reactions. Now, next one. After this, A granulocytes, come on. In A granulocytes, we have monocyte, the largest of all WBC. Very commonly asked question. Which is the largest in the size? Which is in largest in the size? So when worm infection happens, Gautam, we need to take anti-helminthic tablets. Albendazole you might have heard. When any worm infection happens, eosinophil number increases, but eosinophil is not able to clear the worm infection. So we need to take anti-helminthic tablets like albendazole, which is available, right? Next, monocyte. Monocytes are the largest of all the WBC. Kidney-shaped nucleus. Which shaped nucleus? Kidney-shaped nucleus. And they are also phagocytic in nature. So if you have noticed, neutrophils and monocyte. Neutrophil is a granulocyte and monocyte is a agranulocyte. They are responsible for phagocytosis. You can say monocyte is like a scavenger. Monocyte is like a scavenger which will remove lot of microbes, your dead tissues, your dead cells, it will remove, okay? So it behaves like a scavenger. They engulf the bacteria and cellular debris. Cellular debris means damaged cells and tissues. And how much percent they make? They make roughly 6 to 8% of total leukocytes. How much leukocytes they make? 6 to 8% of total leukocytes. So if you remember, percentage is very important. Next, last WBC of the day. Last point of the day, guys, and then you are free. Last, agranulocyte is lymphocytes. Lymphocytes are how much percent? Lymphocytes are 20 to 25%. So can you tell me the percent very quickly after this? Let's see how much of your people remember the percentage. Lymphocyte is 20 to 25%. There are two major types. The one which develops in bone marrow, that is B lymphocyte. The one lymphocyte which develops in thymus gland, that is T lymphocyte. And both are responsible for immune responses of the body. And if you remember in acquired immunity, I told you T lymphocytes will give you cell-mediated immunity and B lymphocyte will give you antibody-mediated immunity. Okay? So lymphocyte also helps in immune response. Okay, everyone is helping in immune response, and that completes your WBC. Now quickly tell me the percentage. Tell me the percentage, guys. Let's see how much of you remember. Tell me the percentage. Come on. First is neutrophil. Neutrophil, how much percent? Neutrophil, how much percentage of total WBC? After neutrophil, tell me about eosinophil. Then basophil. Then monocytes. And then after this, tell me about lymphocytes. Tell me the total percentage. How much is eosinophil? Very good. Eosinophil is roughly 60 to 65% of total WBC. Okay, next. Eosinophil is how much? Eosinophil is roughly 2 to 3% of total WBC. Basophil is 0.5 to 1% of total WBC. Monocytes will be 6 to 8% of total WBC, and lymphocytes will be roughly 20 to 25% of total WBC. So you can see neutrophil is number 1, number 2 is lymphocyte, then we have the third place of monocyte, and then we have the fourth one to be eosinophil, and the least number of WBC, fifth one is basophil. So we can have order also. We can have the order. Neutrophil number greater than lymphocyte. Lymphocyte number is greater than monocyte. Monocyte number is greater than eosinophil, which is greater than basophil. So like these questions can be asked. You can be asked a match the column question. You may ask the percentage from increasing to decreasing order, decreasing to increasing order, or basic functions can be also asked. Okay, so that is all about WBC, guys. So here we complete the RBC and WBC. Platelets we cannot do, so platelet I will not ask in the exam. So whatever we have studied, lymphocyte, we have studied the immunity concept, innate immunity, acquired immunity. We studied the blood plasma, RBC, WBC is done. Okay, so that's all for this class, guys. Thank you so much, and enjoy your holidays, enjoy your vacation, and see you after this short break in the offline classes. Okay, bye everyone. Thank you for your patience. You gave your so much of time to me. Lot of time you provided to me. So that's it, guys. Bye bye. Take care. God bless you all. Keep rocking.